to have Dr. Thomas Carsey, who's a physical chemistry PhD. He's going to talk to us about just beyond the shore observations of Florida's coastal ocean. And I will turn it over to you.
All right. Thank you. Thank you for inviting me. I believe in citizen science. I'm glad to see a large crowd here tonight. All right. I'm Thomas Carsey. I'm with the NOAA, National Oceanic and Atmospheric Administration in Key Biscayne. And here I'm going to talk about some measurements that we've been doing on the southeast Florida coast. And our co-workers here include Jack Stamatis, who's not feeling well tonight, who runs our ocean acoustic current measurement group, and Chris Galliano, who is here tonight, and with his Mary Beth Gidley, head up our microbiology program. And so with that, next slide, please. As I mentioned, we have a very dynamic program looking at various aspects of the physical and chemical and biological aspects of Southeast Florida. And in fact, many of these people would make excellent speakers for a program of this nature, including the South Florida program, which has been around for many years, looking at Biscayne Bay and Florida Bay, especially with salinity and temperature, and quite an important aspect because with the changes in the hydrology of southeast Florida, trying to regain the traditional hydrology of Florida before the Europeans came in and put dikes everywhere. This changes Biscayne Bay, but they need to know what these changes, the ramifications of these changes, and that's what the South Florida program is looking at. The microbiology program looks at not only the coastal ocean, but also the beaches. They're very active in the Healthy Beaches program and other programs that look at the health of the area. Ocean acidification is a very dynamic program. It's fairly new, and it has to do with the changes in the ocean that occur when you have increased carbon dioxide into the water, which changes the pH. And that has all kinds of ramifications with respect to what happens to calcium creatures in the ocean, which is coral reefs and just about everyone. The Cruise Icon program establishes monitoring stations in coral reefs around the world, especially the Caribbean and in the Pacific. And this is a program which not only interprets, it gathers data from these stations around the world, makes it accessible to scientists around the world, but it's part of what they call an expert system in which they try to write algorithms based upon the data they receive to predict whether like a bleaching event is going to occur in a coral reef, so that divers can be sent out to that and check the location and see what's going on. The Gulf Stream Medis program, headed up by Lou Gramer, looks at whether you can bring high nutrient water into the coastal ocean from the Gulf Stream based upon eddies that exist in the Gulf Stream as it wraps around Florida and goes up the coast. Nutrient measurements, Dr. Zhang has been pioneering in developing new methods, because if you're looking at like chemistries of the deep ocean or the blue water, the concentrations in the surface in the middle of the ocean can be 1,000 times less than they are in the coastal ocean. So it requires very special techniques to do these measurements. My program is the Florida Area Coastal Environment Program up in the upper right, so we'll be talking about that today. We also work with Jack Stamatis on the Inlet program, which looks at what's coming out of the coastal inlets into the coastal ocean. And the Mary's program by Chris Kelbel is a very novel approach. He gathered practically everybody who does science in Southeast Florida, coastal science, and he, with the idea that you could bridge between the scientists who are gathering the data on Southeast Florida and the managers who are having to make decisions about infrastructure and changes in the way that the area is constructed for human use, to match those two together. So that if a person wanted to, say, build a pier, you could answer the question of whether this pier would be economically viable in the sense that it's going to increase the value to fishermen and boaters, but decrease the value to someone who would be looking at the biological aspects. Anyway, very interesting program. So, next slide. What we want to do in Southeast Florida is to maintain to maintain at least the health of the area the way it is now, if not to make it better. And that means we want to maintain low concentrations of key nutrient chemicals in the coastal ocean. Now, nutrients that I'm speaking of, nitrate, nitrite, ammonium, phosphate, and silicate, are the things that phytoplankton, that plants need to grow. Just the same things you put in your water to water your plants with. But in fact, we don't want a lot of nutrients in the coastal ocean, unlike your lawn, which you do want a lot of nutrients in your lawn. Because if you have too many nutrients in the coastal ocean, it leads to eutrophication, a disruption of the balance of the processes, and in the case of the coastal ocean, can lead to explosive growth of algae and a disruption of the food webs and all kinds of terrible things we don't want. So we want to keep the concentration of nutrients low. This is called oligotrophic. And we do that by trying to limit the amount of nutrients and other chemicals that come out of the continental landmass into the coastal ocean. We typically think of this as being of two kinds: point sources and non-point sources. Of course, the point sources are the places where there's a specific place you can say, here's where it's coming from. Specifically, coastal inlets and treated wastewater outfalls. We have a treated wastewater plant on Virginia Key, the next island. And the effluent from that goes out into the coastal ocean, and people want to know what effect that is. In fact, it's a really hot topic. We'll talk about it in a minute. Non-point sources are a lot tougher to measure. You know, it's pretty easy to find out what the treated wastewater plant is putting into the ocean because they monitor it, they measure it all the time. But how much stuff is coming out of the inlets? And how much is coming out of city runoff? And groundwater is a really difficult measurement to make. It's going through the groundwater and coming out into the coastal ocean somewhere. That's a very difficult measurement to make. Ocean upwelling is a means by which you can take deep water, which always has high nutrients, and bring it up to the reefs. Very important business. And advection refers to water, for example, that comes out of Gulf of Mexico and rounds Florida and goes up into our coastal ocean. That can happen. So, next slide, please. Well, this has been a pretty hot business in some cases. I don't have my clicker here, but there's a lot of different issues that have occurred that have gotten into the news. And it's kind of entertaining to look at the history of that. There was a big thing a couple of years ago with something that some algae growth in Gulf Stream Reef up off of Palm Beach, which ultimately led to a law which said that all the treated wastewater outfalls would be shut down by 2025. And that's not the only issue. We've also had issues with respect to the port expansion and the dredging and the sediment that comes out of the dredging and how that affects the coral reefs. This is a big deal. And right now it's in the news a lot because of what's happening with Miami. The EPA was sued and to establish new standards for nutrients all over the state, and they've gone to a new kind of standard called numeric nutrient criteria standards that exist all over Florida, and that's also been in the news a lot, and it's very controversial. So we found ourselves in the midst of all of it with this program. The treated wastewater business, they did modify that law 2 years ago, which allowed treated wastewater plants to use their outfalls during times of excessive flow. It turns out that when it rains a lot, water actually goes into the sewage system and becomes a problem with exceeding the capacity of the sewage treatment plants to process it all. And that's when they want to use the ocean outfalls. They're leaky. Have a seat. Next slide. So today, what I'd like to do is talk a little bit about physical oceanography of our coasts, something about how the water flows. And then I'll talk about some chemical studies that we did in a program off of Broward County. And I'll briefly touch about microbiology, but I would invite a more in-depth discussion of microbiology by inviting Christina Allen to give a talk here at some time in the future. And then I'll briefly talk about our project with the Florida Department of Environmental Protection to generate numeric nutrient criteria standards for the coastal ocean. Turns out to be a lot tougher than it is if you're trying to do a lake or a river. Next slide. Oh, before that. Over on the right is a map showing the area that we're talking about in Florida, and I put on there those little triangles are the locations of the treated wastewater outfalls. Now they're the ones who have been partially responsible for funding this project. However, we have endeavored to make sure that we follow good science in working out this project, and we're not taking any sides on the business of ocean outfalls. We just want to know what's out there. So, the Gulf Stream. You know, we all know about the Gulf Stream. We see little arrows drawn on the maps. The Gulf Stream is called a western boundary current. And as you can see, where's my pointer? The Gulf Stream is in a loop. If you see in South America, there's a parallel loop going the other way. And the same is true in the Indian Ocean and the Pacific Ocean. And these are all driven by wind, which blows from east to west along the equator and causes these gyres to exist. And the little arrows that you see for the Gulf Stream is kind of a lie, because that's a long-term average. If you go out there on any particular day, it wiggles. It's kind of like if you throw a Frisbee. And the Frisbee doesn't just go in a straight line. It meanders all over the place. And if you had a Frisbee that was this big, it would meander a lot more. And in fact, this is a problem in fluid dynamics. It would make a great science fair project, by the way. You take a flow of a moving liquid next to a boundary. Now, the liquid right next to the boundary has zero velocity because it's right next to the boundary. But what happens in the boundary in between is you set up a non-laminar flow. And you get all kinds of little twists and turns right there. It's like shown here in the diagram. And this business of the twists and turns is what you're really seeing in the coast of Florida, not the straight line that you get if you're way out on the Gulf Stream. It's a big difference. So I'll show you that in a minute. Especially in Florida, as you can see, Miami, where the distance to the Gulf Stream is very short. It's near. So the effects are very strong. Next slide. It's gone. No, disappears.
Oh, it disappears on the screen.
It's gone. So the way we measure ocean currents is quite interesting. They use a device called an acoustic Doppler current profiler. Now we all know about the Doppler effect, the old thing about the moving train or whatever, and the sound changes as you go by. Now with a Doppler equipment that we see over on the right, ADCP, it sends up beams of acoustic beams into the ocean. And it turns out these are reflected back by little particles in the ocean that are moving around with the current. And the time that it takes to come back to the instrument, to bounce back, to be reflected back, is proportional to the distance. So by taking several beams, as you can see on the right, and doing the right kind of mathematics to these beams, it can compute an X, Y, and a Z motion in different, what we call bins, different little units of distance up from the instrument. Amazing. This instrument just revolutionized physical oceanography because you could get such accurate flows. Before they have these little spinning devices and all that. It was just a mess. So here's two of them that we have in the Broward experiment. You can see the unit is inside, it's cased in this block of concrete so that it doesn't move around during current flow. We've placed a dozen or so of these at different times off the southeast Florida coast for different experiments for different purposes. And occasionally we were smart enough to actually place two of them working at the same time, but most of the time it was just one. So we'll be talking about the measurements that we made with these instruments. Next slide, please. these, the presentation that I have, or the upper one, the little blue spikes represent where the current is going at that particular time. So down at the bottom you see day 225. So up is about maybe day 230 or so in the year, and the spike means that the water is moving north. Now meteorologists have it all messed up. They point the arrows where the wind is coming from, but oceanographers will tell you where the ocean is going. A big difference. So here we are, the ocean's going north. Now the top bend is about 6.7 meters down, it's near the surface. And then as you go down 13 meters, 21 meters, 29 meters, that's at the bottom. So these are the different bends that we have in the coastal ocean for about 4 months in this unit that you can see that's up there where the red arrow is. Now, there's a lot of amazing things about this. One of them is, as you go down, the directions are all the same. So whatever happening at the top is happening all the way down, just slower. Because when you get down to the bottom, the velocity is 0 because of drag. So amazingly enough, whatever you see happening is happening in the entire water column at the same time, practically instantaneously. And look how often it changes from going north to going south. I mean, this is moving a lot, and the entire ocean is moving back and forth. The whole thing. That's amazing. The whole thing. Now, that's energy you could capture, it seems to me. And you wouldn't have to go that far offshore. You wouldn't have to go to the Gulf Stream. This is right here. This is close. So we see a lot of reversals in 4 months. We get this similarity all up and down the water column. And there's very little time spent in east-west motion. It's always either north going south. It's just goes very fast between the two. Next slide, please. So here's another one. This is located down near us. This is the mom's site down south, the red arrow. And here I plotted these 5, 6 different months. And I wanted to show you the variation that you get in the currents. Here in July, for about 3 weeks, the water's going exactly the same direction, change the speed, but it's all going north, the whole thing. Now this is only one depth this time, and different months as opposed to different depths. But you know from the previous slide that during that time, all the water is going north, all the way up and down the water column. Following that, there's this incredibly rapid shift. Why? We've been trying to figure that out. It's like this random motion, these flips that you see when you have the liquid flowing next to a boundary and you get all this turbulence. This is basically a turbulence signal. Just amazing. So I wanted to show you how this can, from month after month, you get these situations where you have flow going one direction and then it flips, and then it can flip very fast. So, on occasion we find that the southern flow is associated with wind from the north, but most of the time there is no correlation. There's no correlation with anything. Next slide. So here, we happen to have 4 different ADCP units operating at the same time. Up here, next to the Gulf Stream Reef, up near in Palm Beach, a place that was highly controversial a few years ago because it was a place where there's a lot of algal blooms, and so we did some studies up there. So at the top, Gulf Stream Reef North, they can see on the right where that is. Gulf Stream Reef Mid, which is actually inshore. Gulf Stream Reef South, south end of that reef structure, and Boca Raton. And once again, you see, particularly on the 10th or so of March, the very same things happening over a pretty wide range of the ocean. And they're happening at almost exactly the same time. In fact, the difference between the timing of this, whatever happens at the bottom happens at the top about half a day later. So it's very fast, and it happens all over, practically instantly. Just amazing. Next slide. Now, last one. Here I've got some instruments that are located east and west of each other. So I want to find out what happens inshore versus what happens offshore at the same latitude. Yeah? And there, the top 2 are two ADCPs, one run by Hazen and Sawyer, which is out at depth at about the third reef track, and the lower one is only out like very close to shore. And again, you can see the same kinds of things happening, which means it's happening inshore and offshore at the same time. But in fact, it happens inshore first, and then happens offshore. And the difference is about 45 minutes. Not only the graph on the left shows you that wherever you are in the coastal ocean, if you wait 8 to 10 hours, the water switches direction. I think that's just amazing. Next slide, please. Now here we compare two ADCPs at the north end of Broward County versus two ADCPs at the south side. And at the top, the zero line shows you that most of the water goes north most of the time. The blue that you can't see, it's kind of behind the orange, is the inshore, and it shows you that it's more evenly distributed. But if you get down to Hollywood, the nearshore is in fact going south more than half the time. So the idea, and you hear this a lot, that the Gulf Stream means all the water is going north almost all the time. That's really wrong. And in fact, off of Broward County, it goes south near shore more than half the time. Now that means water coming out of the inlet is going to be going south before it goes north. And that has a lot to do with what, if you were around an inlet, you may want to know what's going, you know, this is the way it is. In fact, I have a picture down here of the Boynton Inlet over in the bottom right, and that's everything's going south. And a lot of Google Earth images of inlets actually show things going south. Anyway, next slide, please. Well, these ADCP measurements devices also have temperature measurement devices on them because velocity is related to temperature. So we have these temperature measurements from way out in the ocean, and this red, in the red one, you can see, the main curve here you can see is just summer and winter temperatures. But look how in the summertime you can have these gigantic temperature drops of 10 degrees. This is at the ocean floor. So something major is happening to drop the ocean in 10 degrees, temperature drop. These things may exist for a couple of days, 3 or 4 days, and then it goes back up to what it should be. What's happening? Well, we think it's upwelling, which is a device by means you can take Gulf Stream deep water and bring it up to the third reef tract and maybe into the second reef tract, which brings a lot of nutrients to those reefs. And we don't want nutrients on the reefs. And those are natural phenomena that's been happening forever. But we need to quantitate that and measure it and find out what's going on. And a lot of physical oceanographers are very interested in these processes, and they have not been studied. And they're waiting for somebody to come along and study them. There I did a contour plot of the temperature in one of these events that we happened to be there with our ship on that, where the arrow is, and took some measurements and did find a whole bunch of excess nutrients at that point. Next slide, please. So we did this experiment off of Broward County, we did a year's worth of cruises, 24 cruises in the north end of Broward County and in the south end. We did all these chemical and physical measurements you see on the left and on the right there, and Chris did the microbiology that you see listed there. So I'll review some of those results. Next slide. It's a lot of fun to do these because you get to go out on these small boats. The major boat we use is the Hildebrand over on the right side. It's a former U.S. Coast Guard utility boat, twin diesel, lots of fun. We take interns, by the way. It has on the side, the back, it has an A-frame which allows you to drop a device in the water called a CTD, which is what Rachel and Chuck have on the left. Now, the CTD stands for conductivity, temperature, and density. And with those, you can compute all kinds of things. And the instrument, which is in the center of that, the CTD instrument, has a bunch of other probes on it, like turbidity and things that you can measure. And then it has 6 4-liter bottles on the side, and you open them up, and you drop it down into the water, and then you close off 2 of them at the bottom, and bring it up, and close off 2 more in the middle, and 2 more at the top, and you've got these 3 samples from different depths. They use the same device out in the deepest oceans. It could be miles deep, and they have like 24 bottles, and it takes hours to get down to the bottom, and they capture water all the way up the water column. Just amazing. Anyway, CTD. We also use a little RV cable, a 21-foot Parker. We use it for diver support because divers have to go out and jump in the water and service the ADCP units. And it also has an ADCP mounted on the boat that looks down, and you can look at the currents wherever the boat goes. Okay, next slide, please. So here's some results. Okay, I've plotted up here. The horizontal is latitude, basically all of Broward County, from 26 degrees pretty much to 26.3. It's related to the red arrow you see on the right. But actually those results are very similar to other results we found. Basically the entire coast of Florida is going to be the same. We just have good measurements here. So that's the x-axis. We have dissolved inorganic nitrogen at the top, which is the sum of nitrate, and ammonium. And what you see is very low levels most everywhere except around the Hollywood Outfall in red, the Port Everglades Inlet in blue, the Broward Outfall in red, and the Hollywood Outfall in blue. And I have a line, you can barely see it, at the bottom. That's the background average for surface, mid, and depth. Surface, red, mid is blue, and green is deep. If you go down to phosphate, very similar results. Very low background levels, and these big peaks right around the inlets and the outfalls. Same with silicate, another important nutrient. Yeah? And then down the last one is turbidity, which has to do with how light can go through the water. Now, if you want your reefs to be healthy, they have to be able to see light. And they're down about 30 meters, the third reef track. So light has to penetrate all the way down. You can't do that if you have turbid waters. These are very clean. Turbidity of less than 2 is pretty darn clean. The state has a limit of 41, I think it is, for inland waters. Anyway, so we've got clean background conditions interrupted by these really high peaks right around the inlets and the outfalls are point sources. And it looks like if you go away from those point sources a little bit, it goes down very rapidly. Remember the bottom, especially the bottom green lines here, those are where the reefs are. Those are where the divers are. That's what they want to keep clean, and they are clean. And they're low concentrations. And there's no change with latitude. It's not like you've got this river running north and you keep throwing trash in it and the backgrounds keep going up and up and up. We still have no evidence of that that's statistically significant. Next slide. So I'll talk a little bit about ecosystem health. Now, there was a guy named Redfield, and he went out and he got his friends to give him samples of plants and then biological material from all around the world. This was many years ago. And he analyzed all these for basic elements, carbon, nitrogen, oxygen, phosphorus, and life. And he found that the ratios were always the same everywhere he went. That's amazing. That's called the Redfield ratio. It's kind of like the periodic table or something. It's one of the things that you see all over the place. It's true. So that means, for example, carbon 106, that's the number of moles. Hydrogen, oxygen, nitrogen, phosphorus 16 to 1. That means if your water has not enough nitrogen, it might have a ratio of 14 to 1 or 10 to 1. That would be like, and the other important thing is Liebig's law of the minimum. Liebig says that nutrients in the lowest concentration would control the growth rate. That would be like if you had 1,000 people, but only 3 of them were female. The growth rate of that population would be limited a lot by the low number of females. That's growth limited. If you have the proper ratio, in the case of humans, 50-50, then you have maximum growth, yeah? Anything different than that, growth limited. In the case of Redfield ratio, we want 16 nitrogens for every phosphorus. Otherwise, you're limited. Well, it turns out, I plotted at the top some of the water ratios of phosphorus to nitrogen, and I put the TNTP line to 16. And what we have is a situation where we're phosphorus limited, which means in the coastal ocean, you could dump a lot of nitrogen into the coastal ocean and nothing would happen because nitrogen is not limiting. But if you put phosphorus in the coastal ocean, it would mess it all up because phosphorus is currently limiting. Okay? Very important principle. You probably already know that. Dissolved oxygen. Life needs oxygen to survive. In the human body, your blood needs about 97% all the time of oxygen. If it goes much below that, you can be in deep trouble. Our measurements indicated almost every measurement we took was nearly 100%. Now, if you have eutrophication, like you drive along and you see this lake and it's yellow, it's green, it's all messed up, that means it's a case of eutrophication, excessive growth of algae. The algae all die, and in the process they use up all the oxygen in that lake, and everything dies after that. So high oxygen levels mean you don't have that situation, you have a healthy system. And we have a healthy system, practically every measurement is 100%. And then chlorophyll. We have a measurement of chlorophyll, which is an indication of plant growth, but we have another measurement called phageopigments, which is a measurement of dead chlorophyll. So we would like to have life more than death. Yeah, we want to have the concentration of chlorophyll a lot more than the concentration of phageopigments. That's another sign of a healthy ecosystem, more life than death. And that is the case, it's running about 3 to 4 to 1 in favor of life. Excellent. We also see no relationship with nitrogen or phosphorus, indicating you don't have problems with excess nitrogen or phosphorus causing explosive growth of chlorophyll. Anyway, next slide, please. Now, we were really interested in these outfall plumes. Treated wastewater, it's treated, it's not untreated, which means it's been filtered and chlorinated. And there's a plant right here in Virginia Key. Then it goes out of a pipe, usually a couple miles long, at the edge of the Gulf Stream, at about the 3rd reef track. And these lines are in the ocean floor. It comes out and it has no salinity in it. It's fresh water. It immediately goes to the surface. It's buoyant. And so we were interested in whether this plume was having a negative effect on the environment. So we put a bunch of sampling stations with our boat around the outfall, north and south, and one right at the outfall. And then we measured for all kinds of important things. There they are. Now what I've done is I've plotted, each one of those is plotted with distance from the outfall, with 0 right at the outfall, and kilometers north or kilometers south. And what did we get? Well, like in the middle, N plus N is nitrate plus nitrite, and that's the kind of nitrogen that would be used by the plants. Very high values, or more or less high values, right at the outfall. But if you go a kilometer or 2 away, it just vanishes. Surface, mid, and bottom depths. You can't see it if you go 2 kilometers away. It's gone. Similarly with ammonium, phosphorus, silicate, and the others, chlorophyll, nothing. Not only is there nothing in the plume, but there's nothing in the plume that's making chlorophyll downstream. There's nothing. Salinity, now these are fresh water, but the salinity, the dilution is so fast that you hardly see any signal even in salinity. Imagine that. Now, the surface salinity is the blue one. It dips down a little tiny bit, mainly because it's such a hard thing to do to catch this boil. If you go out into the ocean, you find the boil on a clear day, because it's not clear, you can't see anything. You may see a boil there, and there's usually a bunch of fishing boats around it. And then you park on the boil, and you bring out your CTD, and you drop it in the water, and you look, and the boil's moved 10 yards away. And that's the way life is. It's It moves all over the place. So catching a boil can be really tough. So, a little bit of temperature effect at the bottom, not very much, a couple of degrees. pH, a tiny little drop, a little bit more, so it again disappears very quickly. No effect on TSS total suspended solids, nothing there. Nothing in the chlorophyll. So, next slide, please. That's the Hollywood treated wastewater outfall. The Broward treated wastewater plant outfall. Again, it's right past the third reef track. Similar kind of results. You can see a couple stations north, a couple stations south. And the results are the same. We get high values of some things right at the outfall and a few kilometers away, and it's just gone. So in other words, we're trying to see an effect on these outfalls, and the fact is, if you're not right there, that's completely invisible in the coastal ocean. It just doesn't, it can't be seen. You don't know you're there. All right, next slide. Now, we wanted to know if the plume comes out of the outfall, it goes back to the surface, it starts spilling out. Does any of it go down to the reefs? The reefs are what we wanted to protect. You know, the surface is not that important. We want to protect the reefs. So what we've done was plotted here. This time concentration is on the x-axis and depth is on the y-axis. So we're going down, and these different colors refer to the different locations, and somehow I split it around so that the Broward locations are at the bottom and Broward concentrations are at the top, and vice versa. So the point is, you can see, except for the red ones, which are the sample right at the outfall, there is nothing when you go down. There's no evidence that something's going down from this plume, down, lower down to get to the reefs. Nothing. Okay, next slide. Well, we went to the inlets and tried the same kind of experiment. Yeah? So here's Port Everglades, right up the road, and Hillsboro Inlet, further up the road. Now 0 here is right at where the inlet goes out into the coastal ocean. So the negative values refer to the lake or lagoon behind the inlet. Okay, that's going to be high. But and then I plotted these other stations plotted by linear distance away from the outfall, or the inlet. Okay. Once again, as you go out a kilometer or two, everything drops back down just to the coastal ocean. Within 2 kilometers, you can't tell it's there. It's gone. The same that we saw with the inlets. I'm sorry, with the outfalls. Now the dotted lines refer to this background concentration that you get away from the inlets. Okay, next slide. Now, during those same samples that I was taking chemical measurements, Chris over here was doing microbiological measurements, which he could talk for hours about, and he will if you let him. But the point is that we found the very same effect. We found enterococci was, at the outfall, was very high, and when you went away from the outfall, it dropped rapidly back. Now these enterococci are dead or dormant because outfalls are chlorinated, but inlets are not. And that makes a big difference. Everything is dead. Next slide. So we did these experiments with dye. We have this rhodamine dye, which is really, really deep red, and we went to the treated wastewater plants, and you put this into the container just before it goes out into the ocean. So you can see from the Hildebrand, there's a shot of the red dye on one of the experiments that we did off of Broward. And just to show you how it is to do these experiments, we went out there and placed these, on the right you see the map with the M1, M2, M3. We put these moorings out, which are weighted down with some railroad wheels, and then there's a line that goes up to the surface, and we put an instrument there near the surface where we measure various things, including the dye. We put them north of the outfall, of course, because we know that most of the time current goes north, yeah? So there we are. And then then the red, on the left, the red triangles are where we did the drops of the CTD with the Hildebrand, and you can see, yes, the current was going north at the very beginning, and 10 minutes after we got there, it switched and went south, and all of our moorings were useless. Because we didn't think enough to put one of them at least south and two north. No. Anyway, so we only saw a little bit of diet in one, and that's it. We didn't see anything. Next slide. So on the Hildebrand, we had this method where we could bring water into a water intake and then bring it on to the deck and measure the rhodamine dye, and wherever we went, then it was measuring dye. And the little blue dots are where we saw maxima, little peaks in this rhodamine signal. So we knew there was dye there, yeah? And then we stopped occasionally and dropped the CTD so we could get rhodamine concentrations down in the water column. And then we had the moorings, of which only one was any good. Now, on the right, I've plotted the rhodamine dye concentration on an x-axis and depth. And with the exception of that one point, the only time we saw rhodamine of any kind of concentration at all was at the surface. We never could find any dye that could go down, just like with the nutrients, the chemistry. It just didn't go down. There's no way that I can see that anything from that plume could get down to the reef. I mean, we tried to find it, and we could not find any evidence that it ever could get there. Next slide. And here's some of the data that shows it's a very typical dilution diagram for a plume with a log demonstration there of the dilution. Dilution values were very typical. And on the right is a contour plot. These computer programs are so cool because they can take all this data and they turn it into a contour plot. And the little yellow dots are where we did the measurements, and then the computer generates this contour that shows you the shape of the plume as a kind of a weighted average for the whole experiment. And you can see how it's elongated mostly north and south because of the current flow. Okay, next slide. So, the numeric nutrient criteria study is a new project. We're about 1 year into the field program, 1 more year to go. And here, the state of Florida wants to set a level of nutrients so that if you're above that, there's some kind of demonstrable effect on the ecosystem. But if you're below that concentration, you're okay, everything's fine. So, we want to take simultaneous measurements of the water chemistry and the coral health, so that if you see an exceedance and nothing happens to the coral, then you know that you're within this good range. But if you see an exceedance in something and the corals begin to degrade in some fashion, then you know you've passed the boundary. You can set these criteria in theory. So here we have gone out 6 times. The data on the left there, in the bottom, sorry, represents the surface, mid, and depth concentrations for these 6 experiments during these 2 reefs that are pretty close to here. That is Emerald Reef down south and Pillars Reef a little bit north of here. We may have dove on those reefs. They're nice reefs, apparently. Anyway, so what do we see? Looking at the middle ones, the total nitrogen at Pillars and below that total nitrogen at Emerald Reef, it looks like these are going up. So that's interesting. We might see if we can pass some kind of threshold. Total phosphorus is about the same, and this is true whether what level, what depth you're on, they're all about the same. But the divers tell me that the coral reefs are extensively bleached in these 2 areas, more so than the 2 areas that we go to in Broward County. And the reason they're bleached is because of what we see down here on the left, excessive high temperature. And this is the way the world is. This is high temperature, and you know where it's from. Well, in fact, in this case, it's from Biscayne Bay. Because Biscayne Bay, too bad this doesn't work. Of course, down at the bottom there, it's not easy to see. Biscayne Bay is shallow. The water heats up a lot because it's not getting the fresh water that it used to get in pre-Columbian times. It heats up and it flows out. It gets caught in the northward current and it bathes those reef tracts, and the water is really hot. And the people on the boat talked about bringing up the CTD and open up the sample, and the water is just hot. I mean, it's like taking a bath. This is water that the reefs are exposed to. This is what we find. Look at those temperatures. They're really high. Surface, mid-depth, and they're all high. Now, if that continues on long enough, everything's dead. Now, it probably won't because we're getting into wintertime. So they're going to go out here in the next month or so and they'll see what's going on. Hopefully it's cooled down. But this is really near the tipping point. It has nothing to do with chemistry. It has to do with global warming and the way the world is. Next slide. So, in summary, talking about the currents, most of the currents are northerly, but we have a lot of reversals. They happen a lot. They can happen very fast. They can happen an average of 8 to 10 hours between reversals. That's fast. And the whole ocean switches at one time. There's an important south-flowing current that has not been well characterized. And it happens, the closer you get to shore, the more you get of the south-flowing current. It has a lot to do with the way pollutants coming out of the coastal ocean are going to be affecting that ocean. And upwelling, or some phenomena very similar to upwelling, occurs a lot in the coastal ocean, and it brings up high nutrient water from the Gulf Stream, and it has been doing that for 1 million years, approximately. Next slide. The water column, there's some dispute about this, but in my view, anyway, the water column is healthy. It's oligotrophic, and it's appropriate for life. The inlets are an important source of contaminants into the coastal ocean. Something is causing that background concentration that I showed you in one of the slides before. And it has to do with a little bit here, a little bit here, a little bit there. That all combines to make this background concentration that you see on that first chemistry slide. However, the inlets, and the same is true with the outfalls, which are also a significant source of a number of contaminants into the coastal ocean, when you're a kilometer or two away, you can't see them anymore. It's just gone. It's just diluted. In the case of the outfalls, it's all diluted in the surface water. Now, in the case of an inlet, okay, are inlets fresh or saline? What do you think?
Combination.
Huh?
Combination.
Combination. Because the water that goes out of most inlets is the water that went in the inlet in the previous tidal cycle, mostly, especially when it's in the dry season. You take inlets like in Hillsboro, or even Lake Worth. Water comes in, and water goes out. And the water goes in, and the water goes out. Meanwhile, there's some inputs from the continent, but basically it goes in and goes out. And the salinity in those inlets is pretty darn close to coastal ocean. Now, there's not exactly coastal ocean, so there's a little bit of buoyancy, but it's basically coastal ocean water. Very good.
It's warmer though, isn't it? Is it warmer in the inlets? Because it's a little more shallow?
That depends on the season, yeah. Next slide. Oh, no, and we didn't see any downward movement yet. Go ahead. So this is the team taken from one of the bigger NOAA ships a couple of years ago. And there's the group, and there's my information. All this stuff is available on the net. Just go to aoml.noaa.gov, and it's all there. Be fun. And that's one of the bigger CTDs there. So next slide. There we go. Now, ready for the questions. Yes.
I notice sometimes walking on the Key Biscayne beach area, there's very little wind condition. There seems to be like a foul smell, like a sulfur. Is that natural scent, or is that some type of leftover? I
The
guess
smell of the sea.
that's natural for the area.
Enjoy it while you can.
My son was thinking it was a massive pollution spill or something. The sewage was coming back on shore.
You'll never remember that it's not sewage going into the ocean. It's treated wastewater. Now, if there's sewage, it came from some boat. It didn't come from the treated wastewater, and I can't control the boats.
Yeah, That sulfur smell is natural. It comes from the decay of seaweed and stuff.
I see.
And a lot of the little mangrove ponds and stuff you have here, it comes from fermentation in the sediment.
I see.
In fact, you can get a pretty good smell over at AOML in their lake.
Yeah.
It's sometimes really rank,
Right there.
yes.
Does that affect the ocean when it goes in and goes out? I mean, that fermentation, when it comes out to the ocean, does that add to all the nutrients and things in the ocean? Does that raise the levels at all?
Well, a minuscule amount, yeah. If you look at Google Earth, you'll see a black material coming out of most inlets. What do you think that is? That's mostly natural material from mangroves. It's perfectly natural, but it looks terrible. It's just the humic substances that are produced by the mangroves.
And even though you get a lot of that smell, a lot of those nutrients get turned over very rapidly. So the system as a whole is pretty low nutrients. And part of that smell you're smelling is that recycling of all those nutrients by anaerobic bacteria and stuff.
That's good to know, I
For
guess.
from some of what I could understand, it seems that the nitrogen is strongest in the more shallow part of your testing area, as opposed to like you talked about plumage is like as opposed to the deeper you get past, then you're not seeing as much of the.
But no, there's really very little effect. If you take measurements and go into the coast with more measurements, you see almost no effect. Where you see the effect is around the outfall and the inlet plumes. Now the outfalls are like these pipes going under the water. And it comes
Right.
up and right there.
And that's where you're seeing.
That's where you see it. There's a boil, and then around that, an area about 1 kilometer or so around that, that's where you see the elevated
So,
concentrations.
but do you think the concern mostly is more in the nursery areas, not to have the plume? Is it not so detrimental to have the plume 2 miles out? Or is, in terms of the sea life, is it more detrimental to see the plume and the algae plume in the nursery area?
Well, I don't think you see the plume in any of this,
No?
anything on the seafloor.
Okay.
We could not find any evidence that it could ever get down. Now, something makes this background concentration. Now, it's low, it's pretty low, but it's produced by a bunch of causes, and some part of that is because of these coastal influences, the inlets and the outfalls and all that, but a minuscule amount. And we're talking about the third reef tract for the outfalls.
So it seems that the biggest threat right now is more the global warming.
Oh, by far.
Then
Now,
the-
we don't know everything that comes out of the inlets.
Yeah.
There could be a lot of stuff coming out. I mean, Chris can talk about the microbiological. I mean, there's pharmaceuticals that are coming out, there's cosmetics, there's a million
Yeah.
things that are in the water. Most of them have not been, and in the outfalls too, they're not removed by sedimentation and chlorination.
The salicides, I understand?
Yes, there's
Yeah.
in fact, we've measured some of those compounds. They make tracers. It's a good tracer. You can measure sucralose, and it's a tracer as good as any. Sucralose is not really biologically active very much, and so it persists a long time. And you can use it as a tracer, and it's cheap.
Yeah, and it's especially good sewage or fecal tracer because you basically excrete it in feces and nothing eats it. So you can detect it at very low concentrations out in the coastal ocean. And the only way it gets there is from sewage.
Yeah.
But
Not
I guess-
necessarily from,
I heard there in some parts they were trying to ban it, certain, the microscopic plastic. Things
microbeads and all that.
like that are in a lot of cosmetic products and things that they were trying to ban that.
Yeah, research is pretty new on that. But we put a lot of trash into the ocean, a lot. I mean, we know about the big trash gyre in the Pacific. What we do to the oceans is really pretty nasty. We have to get to the point where we see the environment as part of where we live and not just some place where we just throw our trash. This is one world, and we live in this world. We wouldn't take our trash and throw it in our living room or our bedroom. Why are we doing that with the world that we live in?
just to follow up with the question on plastics, it's not even just the plastic nanoparticles that are in products, but another big problem is the degradation of plastic in the environment, that it doesn't break down completely, but it breaks down to very small particles and fibers. And that's being seen everywhere now. There's a lot of studies where it's beginning to see it in like oyster tissue and filter feeders. Even in a lot of the water samples that we filter for biology, we're more and more seeing all kinds of weird plastic stuff coming up on our filters that we're trying to extract. Lots of small plastic fibers, things like that. Those small plastic particles are becoming very common in coastal water all over.
The manufacturers of these materials don't have any responsibility for the ecological consequence.
Right.
That's not included in the price tag that you pay. So we go and get a can of Coke, and we drink it, and we give it a toss. And it stays in the environment for a very long time, because the price to the environment is not included in the price that we pay. That would change everything.
The consequences are not entirely known, but if you look in the scientific literature, there's more studies suggesting it has reproductive problems, it's causing problems in shellfish. Unlike a lot of other kind of chemicals and contaminants that shellfish can remove from their tissues, they seem to have a lot of problem removing plastics from their tissue. And so this is beginning to accumulate also in our seafood.
Yeah.
And where it goes from here is not really clear.
So for young people, I would say it would be motivating to think of an invention to replace plastic. If
Yes.
that's possible.
Better
Yeah.
plastics that can degrade.
Yeah.
And then if they were to tack on the environmental cost to plastics, suddenly there would be this big rush to find plastics that had none of these negative effects. And do you think they could find
Yeah.
such a thing? Yes, in about 10 minutes.
Right. Very interesting, yeah.
In fact, there's really a lot of great things. We have young people that go on our boat, interns and all that, and they really love it, because you're not only on the water, on somebody else's boat, right, for free, but you get to take measurements and discover things that have never been known before. It's not like downloading a YouTube that everybody else has. This is completely new.
And where do you go when you go out with a boat? What areas do you take the boat out?
Well, we take right now, well, different places. We did this experiment off of Broward County, as I mentioned in the slide, we have all these stations up and down Broward. Right now we have this other experiment where we're looking at areas around coral reefs in Miami-Dade and Broward County. But every couple of years it's a different deal. And there's other groups that are diving out in the Keys. There's a lady right now that's in the Pacific Ocean in a place that's very pristine, taking a bunch of measurements. There's places in the Pacific where you have underwater vents that increase the carbon dioxide concentrations in those areas, very much like they expect them to be everywhere. So we want to find out what survives in high CO2 environments. And there are those out there. And it just takes somebody to go and get there and do the measurements. Now, it sounds exciting to me to go to someplace in the South Pacific and do a bunch of dives in the most beautiful, pristine areas you can think of in the whole world. Well,
Tell them how you started in your field. Tell them that.
how did I start in my field? Well, I didn't start in this field. I moved into it because I wanted to be involved in the environment. And this, in fact, I would say as a young person, take it slow and move where your heart tells you you need to go. And get the background so that at each step you can make the logical movement to the next step. For example, you think you might want to do environmental science, you get a bachelor's degree in some environmental science, and then at that point you could say, well, I want to go into physical oceanography or biological oceanography or whatever it is, and you move into that area and let your heart guide you. Yeah, there's so many great things in the world to do. I mean, it's just unbelievable. So many wonderful possibilities. The world has a dark side. You can't afford to look at that dark side. You have to look at the light, and there's a lot of wonderful things happening out there.
Are there any more questions?
We the artificial reef program, is there any way that that can be used to compensate maybe the warmer areas and establish other reefs to mitigate that?
There's a lot of work being done on trying to figure out where these called refugia that are the best places to grow reefs, looking at what kind of reef species are the best to be placed here and there. Right now, there's a big program to move, they just finished it, to move the coral reefs away from the Miami-Dade area of the dredging materials because the sediment was really much worse than anybody thought. And they moved thousands of little reef pieces to these trees that they put in off of Broward, where they could grow these little reef guys all over the place. And anyway, there's a lot of work being done in that kind of thing. What you don't want to do is put a bunch of old tires in the coastal ocean, hoping that there'll be places for coral to grow on. That's not a good idea.